Thank you, Ashley, and particularly thank you, Angela, and all her supporters with the charity for enabling us to be in the position that this is now the third time we've been able to bring parents and researchers and other influential groups in enabling us to do high-quality childhood cancer research in Britain. So I just thought I'd start with this quotation, which I actually heard from a parent when I was in Toronto. So working with Ron Grant, who's also a paediatric oncologist in Sick Kids Toronto, very interested in Wilms tumour, and with Angela, we've actually developed a Wilms tumour parent involvement and research group that has reached out and had international meetings. And one of the Canadian parents actually whose child was at that point going through treatment for relapse, said, "I didn't understand what research is until my child relapsed." And yet the irony of that was that this child had already been on a clinical trial which was aimed at optimizing their first-line treatment, and I believe had also consented to have their child's tissue stored in the hospital's tumour bank for use in biological therapies. Research. But the parent, obviously, they're just passing by at the time of first diagnosis. And also, and I think it reflects the fact that treatment of childhood cancer in the Western world is very much based on evidence. We've always practiced what this so-called evidence-based medicine because we are facing very life-threatening and serious diseases, and we have to give hard, difficult treatments with a lot of potential toxicity in order to beat the cancer. So paediatric oncology as a community has always worked together at a large scale and put newly diagnosed children into clinical trials in order to capture detailed data so that we really understand both the benefits and side effects of what we're putting each child through, and we're constantly trying to refine and optimize how we make decisions for each child. So this is the balance that we face for every patient. And for most childhood cancers now, the survival is very good, and in many cases it's approaching 90%. So you're always trying to balance, can you reduce or back off on the therapy a bit, but then there's the fear of relapse, versus if you give too much treatment, there's more risk of long-term side effects, and also the quality of survival, both during and after treatment, can be affected. And this has really been the mainstay of paediatric oncology since the 1960s, and is effectively personalised medicine in that you take account of everything you know about each child and their tumour, how they respond to the treatment, how they tolerate the treatment, and you may have to adapt a treatment programme as you go along. Now, if I take the example of neuroblastoma, so this is a childhood embryonal tumour typically occurring in very young children, has multiple behaviours, and it can be localised, can be cured by surgery alone. It can be sort of intermediately spread, in which case the outcome very much depends on the genetic changes in the tumour or the tumour biology. If it's already spread, even with our most intensive treatments now, the cure rates are still only just creeping up towards 50%. And then it has a very bizarre subtype called stage 4S for special, which only occurs in very young babies, which can actually spontaneously resolve without any chemotherapy or maybe a single course. But as you'll notice, there's about 120 children a year affected in the UK. About 1 to 2% will have a family history of the disease. So there's multiple subtypes of neuroblastoma, and yet there's very few patients in each category. How do we make progress? Well, we make progress by doing really detailed, accurate and innovative research, but working with partners across other countries. So the first sort of introduction of biology into risk stratification came with neuroblastoma, because we've known about this gene MYC-N, used to be called NMYC, which can be present in multiple copies in a tumour. And the yellow line here shows the survival in a child with localised neuroblastoma, not a child, sorry, hundreds of children, if the gene is normal. And here you can see if the gene is abnormal, there's too many copies, the survival chances are half. But look at the numbers. In this study, there were 1,400 children up here and 26 here. So you can see that the presence of this extra amplified MYC-N in localized neuroblastoma is incredibly rare. This is actually a study from the United States. So Britain would have about a tenth of that number to put into such a study. How do you do research in these circumstances? But we also now know in neuroblastoma, and you may hear this, I'm sure, later from the next speakers, that it's not just MYC-N that's important. There are other drugs, other targets, like aurora kinase is a new drug that can target MYC-N. And then more recently, this gene called ALK has been found to be mutated in neuroblastoma. And what's very interesting for the pediatric oncology drug community is that the same gene is mutated in adult lung cancer. But not in all lung cancers, in a very, very small percentage. But what it means is that the pharmaceutical industry is interested in developing drugs against ALK because there's a big market in an adult cancer. But, how can we harness those drugs for benefit in these children? Now, this is a typical, maybe slightly atypical flow sheet. If you've had a child who's been on treatment for cancer, you'll have been presented with a flow sheet from a protocol. This is for high-risk neuroblastoma. You can see how complicated it is. All of these arrows are time points, drugs, different treatments, randomizations, and so on. How do the doctors looking after your child make a decision about which of these routes your child's going to go down? Well, it's only through doing research and doing clinical trials where we give the best possible backbone of standard of care. And then on top of that, we may compare two approaches, like this is two different ways to give high-dose therapy. We may compare whether a child gets any sort of immunotherapy or not against the sort of standard backbone of continuing treatment that's known to help prolong remission-free survival. So all of these things need a lot of information about the clinical features of a child, its age, the type of tumor it's got, how far that tumor spread, what it looks like down the microscope. But increasingly, and you're going to hear next, biology is playing a major part. So this shows where we've got to in a childhood brain tumors. And Darren will say more about this now, but I just want to make the point that you can use all of these aspects of biology and some clinical features to really group children into different groups of what's caused their tumour, which type of cell it's come from, and increasingly now to do with prognosis, and not so much in glioblastoma, but in medulloblastomoma. And in medulloblastomoma, there is a new target found, a gene called the sonic hedgehog signaling pathway that seems to be involved in medulloblastoma. Very exciting. This is actually a young adult with relapsed medulloblastoma, and this over here, this scan shows all the black spots shouldn't be there. That's the tumours everywhere. He was given this single targeted agent, and you can see everything disappeared pretty much. Excuse me, sorry, I couldn't just get a glass of water. But within a few months, it's all back again. So this just illustrates what we know, is that targeting just a single biological target is not going to be enough. But clearly there's something in there. These things can have an effect on cancer cells, but how do you get that sustained response? Now, we know an awful lot in biology, and it's not just to do with cancer, about what makes cells divide, grow, differentiate. And more now, there are lots and lots of targeted drugs that will try and silence or switch on or off these things that may be going on wrong in a child's cancer. The challenge is that children's cancer is so different to adults. Over here, you've got typical adult cancers. The vast majority are typical epithelial cancers or carcinomas. Here you've got children, so they get leukemias, brain tumours, embryonal tumours, and a lot more sarcomas, and teenagers sort of in the middle. So whilst the molecular targets may be the same here and here, clearly the rest of the tumour is somewhat different. And how are we going to put these multiple drugs together? That is a challenge. The other challenge is the number of mutations per tumour. So again, over here, we've got adult lung cancer, some types of colon cancer, which can have literally hundreds of mutations per cell in the tumour. Over here, you've got the pediatric tumours, where the numbers are sometimes in single figures. And what we do know is that this may impact on, for example, the likelihood that you may respond to immunotherapy. Now I want to talk about, there are other aspects to research which are equally important in maximising each child's chance of survival. So these are the steps in a so-called cancer patient's journey, applicable at any age. And really we are increasingly wanting to do research on routes to diagnosis. How do you actually ensure cancer is diagnosed early, that everybody's getting the best practice treatment choice, that it's delivered in a technically expert way with excellent supportive care, and then the quality of survival is maximised, that patients are empowered to be able to proactively look after themselves and have, react properly to early warning signs that things may be going wrong, and are there indeed treatments that might prevent or reduce the risk of some of the late effects that are inevitable from the treatment that a child may require. Now, the good news is we've got fantastic cancer information in the UK now. And this is Public Health England. Jem Rashbash is the director. And so for every step in this cancer journey now, there's a whole host of data. This is routine healthcare data sitting behind it. The most important, of course, of which is the initial registrations that you're identified as a cancer patient with a data diagnosis in the national system. So why is this important? And you can see we're collecting now data on imaging, chemotherapy. I think ChemoCare are here at the exhibition, so all the electronic chemotherapy prescribing goes into this system now. We can start to look at what treatments children are having at a population level and how that might relate to their outcomes. The first step in making this sort of publicly available to everybody and also research is the National Cancer Dashboard that was just published in May. This shows you what it looks like as a headline message. So they're trying to get in something about what are we doing to prevent cancer, what are we doing to diagnose it early, which is 1 year survival, what are we doing about patient experience, and there will be metrics on quality of life. And you can either look at a snapshot or you can look at trends, and the lines here are the England average, and this is your geography. So these are CCGs, or clinical commissioning groups, which there's 34 of them in London, for example. But what does it look like for children? So for children, there's a single metric. So we've got into the National Cancer Dashboard, so already that's a good thing. The government care about what happens to children with cancer, but it's a single metric, which is 5 years survival from all childhood cancers combined for the whole of England. And you can see that for children diagnosed in 2009, the survival was just over 80%. And also participation in research is something that's going to be collected as a metric at a national level. You can also look at trends over time. So here's the 5 year survival trend for adults. Very smooth because there's just so many adults get cancer, nearly 1 in 2 of the population now. What I just want to highlight to you is how jumping up and down the childhood curve is. So that doesn't mean treatment got worse between here and here. It's just that there are only about 1,200 children a year in the whole country with any form of cancer. The exact mix of those cancers will vary slightly from 1 year to the other. And so this is really just random chance. The most important thing is to look at the overall upward slope of the curve. But it also means that you can't just say compare that point to that point and think, oh, things have got worse, because you have to know what your statistical range is. But the good news is that childhood cancer survival rates continue to improve, and you can see here, even in the last sort of just under 20 years, we've gone from just under 70% to over 80%. So that's very encouraging, and that's for all age groups. And these data are all publicly available. You can go to the Office for National Statistics website or the National Cancer Dashboard. You'll find them all there. You also, of course, need a robust clinical system to deliver both standard of care and clinical trials, and we have that with the CCLG and with our experimental cancer medicine centres and active researchers. But I think it's very, very important that we have parent, patient and public involvement in research. It's known that this does improve trial design, the rate at which patients will be willing to take part in studies, and ensures that the research is asking questions that are most relevant to the people affected by those diseases. And in the UK now, most funding bodies and ethical committees will expect written and documented evidence that the researchers have engaged with the patient community that they've been affected by to get success. And then just as my final slide of content is just to say my next talk, because you're going to get me again after the coffee break, I want to tell you a bit about what we're trying to do at a European level to address all of these 7 aspects of childhood cancer. And again, this strategic plan for improvement is available on the SIOP Europe website. I just want to say thank you.